Hello class, how are you? So today we're going to cover the cytoskeleton, and today's lecture is basically broken down into two parts like a lot of the others. And in the first part, we're going to talk about some of the biochemical properties of this cytoskeleton, and then in the second part, we'll talk more about the cellular mechanisms that this cytoskeleton engages in. So as you guys know, cells are really dynamic, right? They move around, they can change shape. Everything in the cell, all the organelles are really well organized, and those organelles can actually get moved around. And I want you to just sort of think for a second all the movement that happens during the cell cycle. And we'll cover the cell cycle in a couple lectures. But that's pretty dynamic, and including moving all of the chromosomes and pinching off for a new cell. And it's the cytoskeletal proteins that really help with that. And also, if you guys think back to the video we saw of the cell on the first day where we saw the leukocyte transmigration, you can sort of think about an immune cell and how it can migrate to the site of infection. And so in order for the cell to be that dynamic, the actual cytoskeletal filaments have to be dynamic. And they're used for a variety of different processes, as we'll see sort of in the second part of today's lecture. So here's just a really, really beautiful image of a cell. And what's shown here is the microtubules are in green, and the actin is in red here. But there's a third additional filament besides for microtubules and actin, and that is the intermediate filaments, and we'll see what those do in a little bit. One of the intermediate filaments are the nuclear lamins that actually sort of form a cage-like structure around the nucleus. And in addition to the microtubules and the actin, there's actually hundreds of accessory proteins that help regulate both the actin and microtubules, as well as the intermediate filaments. So to give you just an idea of how these things come into play in the cell cycle and cell division, here's a beautiful little schematic of a fibroblast, and the actin is shown in red and the microtubules are shown in green here. And then during cell division, you'll have the nuclear envelope breaking down, and then the sister chromatids are aligned along the metaphase plate, and then you have your mitotic aster on both sides. They capture the sister chromatids and they actually pull them apart, and then the actin actually forms this cleavage furrow which then collapses on itself and pinches the cell together, and then you're left with two daughter cells. And so this isn't the only thing that the cytoskeleton's used for. It's also involved in lamellipodia formation and filopodia, and it also forms these mitotic spindle and the contractile ring. And then in and around the nucleus here is also the nuclear lamins, and then there's other intermediate filaments like keratin and stuff like that that are actually in the cytosol. So here's what lamellipodia and filopodia look like. Lamellipodia you can almost think of as cell rolling, and the cell sort of just gets pushed along. And filopodia actually produces these sort of little projectiles, and this is all mediated by actin. So actin is stained in green in both of these, and the microtubules are shown in red. Just to give you an idea of how dynamic the cytoskeleton is, this is a Drosophila embryo, and it turns out that cell division in a Drosophila embryo occurs around every 10 minutes. And so if you're looking at interphase, the actin and microtubules are pretty diffuse, and then all of a sudden when you start to enter prophase, the mitotic spindles move to each pole, the centrioles here move to different poles of the cell, and then you get the mitotic spindle forming, and you can see that, and it all happens very quickly. This is a time lapse of under 5 minutes. Another thing that is kind of cool is the way cells move around. Okay, so this is actually a great little time lapse video of basically 3 seconds, and this is a neutrophil, which is a form of a white blood cell chasing a bacterium. So this arrow right here points out the bacterium, and this here is the neutrophil. And here you can see the lamellipodia, and the actin sort of stained in red here. And you can see it chasing around the bacterium, and eventually it will catch it and engulf it. So the neutrophils are a form of phagocytic cell. In epithelial cells, which are much more rigid and don't move around as much, the cytoskeleton is also very important, and it helps establish the sort of polarized nature of the epithelial cell. So if this is like an intestinal epithelial cell, you can see here you have the microvillus, the microvilli that are here, and they have actin protruding out into them. And then underneath that you have sort of a terminal web of actin. And then you have these junctions, which we'll talk about when we get to the extracellular matrix. But the intermediate filaments are oftentimes attached to the junctions. Then you have your microtubules in the cell too. And as you can see, microtubules, as we'll talk about in a second, they have a certain amount of directionality, and so there's a plus end and a minus end to them. Now, yeast are also polarized, especially haploid yeast, and you can see the actin stained here, and the yeast is actually forms these little buds during cell division, and you can see that the bud is growing, and the next bud will actually grow right alongside of it. And so the buds for a yeast, the daughter cells, form on one side of the cell, right adjacent to where the last yeast budded off. Now, in order for the cytoskeleton to be dynamic, it needs to be sort of made up of subunits, and this figure really illustrates that well. Okay, so you have basic monomeric subunits of most of the cytoskeletal filaments, or much of the cytoskeletal proteins. Okay, they're basically these soluble subunits, and then they can form these filament polymers by just assembling. And because they're able to go from polymer to single subunit and back and forth again, that actually is what gives them their dynamic characteristics. So if you have a cell, for example, and all of the cytoskeletal filaments are sort of polarized, so the cell is moving and growing in one direction, the second it receives a signal such as a nutrient source, it can disassemble them rapidly and then reassemble them so the cell can then travel to the nutrient source. All right, so this is just sort of an example of how dynamic they can be. Now, the cytoskeleton also has some very important properties, and some of it has to do with thermal stability. Okay, so if you just had a single row of individual subunits sort of connected together, it's not very strong, and they're kind of thermally unstable, and they can easily break apart, and that would be kind of unfavorable for the cell. Plus, you can have removal on one of the ends, and so it turns out that the cytoskeletons are actually in sort of oftentimes bundled together. And I like to give you guys this quote. This was from Tecumseh, talking about how all of the Native American tribes, if they joined together, they would be much stronger. And he used this example where, you know, a single twig breaks, but a bundle of twigs is much stronger. And he was trying to use that as an example of how all the Native American tribes should sort of come together. And you can sort of see that here, where if you have a bundle of fiber where you have, you know, many protofilaments together, they're a lot harder to break. You can add in loose things on the ends, but, you know, clearly this isn't going to break as easily. And you guys can all sort of, I think, appreciate that pretty well. When it comes to intermediate filaments, these actually have more rope-like properties. They're sort of staggered, and they have basically lateral crosslinks that help to, you know, connect them, and they're much more mushy and can twist much better than either actin and microtubules can. So the intermediate filaments are, you know, are much more malleable and can bend. Okay, here's two very important concepts that I want you guys to know. It's nucleation and polymerization. Okay, so typically if you have individual subunits, in this example they're going to use the actin subunits, there's a rate limiting step where they basically have to oligomerize, all right, and sort of come together, and that sort of creates a seed for the filament growth. Okay, and so you can grow on both the plus and the minus end of an actin filament. And eventually you're going to reach a critical concentration where basically you hit equilibrium, where the addition is equal to the subtraction, and you no longer get overall growth. Okay, so there's nucleation, which is the first step, and then there's polymerization with growth, and then you sort of hit the steady state where the filament stays the same length, and that's all dependent on the concentration of the actual individual actin subunits. Okay, now if you have a nucleation event, you don't have this sort of lag phase here where you have to get the nucleation event to form the oligomers. Oftentimes you'll have a protein here that will help the nucleation, and you go immediately into growth. And so you're basically polymerizing the fragment, and then you'll still eventually get to the steady state where the addition is equal to the subtraction, and there's no net growth in the length of the filament. Okay, so just know the critical concentration is the concentration of subunits where the filament no longer grows, and the critical concentration is basically when the on rate is equal to the off rate, or those are equal to 1. And ultimately, the nucleation event, especially when you're in a cell, that's actually catalyzed by proteins, and we'll see what those proteins are in the second part of today's lecture. Okay, so here's the structure of tubulin, which forms microtubules. Okay, it contains two subunits, a beta subunit and an alpha subunit, and these both bind GTP. So alpha and beta both bind a GTP molecule, but only the beta subunit hydrolyzes the GTP. Okay, and these are basically formed into microtubules in these sort of, you'll have one protofilament, and then when they come together they form a microtubule. And there's actually 13 protofilaments that make up a microtubule. And they're sort of shaped like this. And here's a nice electron micrograph of the microtubule, and here's a cross-section of it showing the 13 different filaments. And now, because of the way the beta and the alpha tubulin are together, it creates a plus and a minus end, okay? And the plus end is where the beta is on top, and the minus end is where the alpha subunit is sort of on the bottom, okay? And so there's a plus end and a minus end. And this becomes very important for plus end directed growth, as we'll see. Actin and actin filaments, they also have a plus end and a minus end. And here's just the structure of an actin monomer, and it binds both ATP and ADP, and actin is actually made up of two protofilaments sort of wrapped around each other. Okay, and here's a nice EM of the actin. And the minus end is sort of on the cleft here, and the ATP binding cleft, and the plus end is the other side. Okay, so just in general, microtubules and actin, because they have a plus and minus, and that actually makes them polar. Okay, and the growth and the shrinkage of these often occurs mainly at the plus end. Okay, now you can get some minus end growth in vitro. However, in a cell, these usually are nucleated down here at the minus end, and so most of the growth is occurring at the plus end. Okay, and just as a reminder, the beta subunit of tubulin is on the plus end, and the ATP binding cleft is actually on the minus end in actin. And what ends up happening in a cell is the polymerization and depolymerization of these, and we saw this in the schematic just a few slides ago, are what are used to do useful work, right? So they can assemble, so the cell can travel in one direction, and then they can disassemble and reassemble on the other end of the cell to cause it to move in another direction. All right, so this whole thing with polymerization and depolymerization is very, very important for a cell to do some useful work by assembling and disassembling them. Okay, so here's another sort of concept I want you guys to understand. It's called treadmilling. And the way basically treadmilling works is in the cytosol, you're going to have most of the actin and tubulin in the GTP bound form or the ATP bound form. And here it's called the T form. Okay, and you know, when you start to add those together, the nucleotide triphosphate form is actually what assembles. Okay, and then it starts to undergo hydrolysis, where the GTP and the ATP are hydrolyzed. And it turns out that addition on the plus end is faster than hydrolysis, so you usually have a plus end cap. Addition on the minus end is a little bit slower, and so you tend to get hydrolysis to the point where addition is much slower. Okay, and so eventually you'll get to the point where more subunits are being added on to the plus end and removed from the minus end, so that the overall length of the filament doesn't change. However, it can actually sort of move along because you're getting addition on the plus end faster than hydrolysis. Okay, and you can sort of think of that here because you get growth on the plus end and sort of slower growth on the minus end. When there's actually no net change in the length of the filament, that's basically the concentration that you'll have. Okay, so in the treadmilling range, the concentration of the T end is actually higher than the concentration of the D end, or the GTP or ADP bound forms. Okay, and the treadmilling concentration is where the length of the filament does not change. Okay, this is the elongation rate. So if it drops below that, it's going to shrink. If it goes above it, it's going to actually grow. Okay, I hope that sort of makes sense. This might be a little bit easier when you sort of see it on this slide. So here is basically an in vitro reaction where they have rhodamine labeled tubulin. Okay, so that was actually added to it, and it gets incorporated right where this little red arrow goes. And so what ends up sort of happening is you're losing subunits, net losing subunits, not technically losing subunits, because so you're getting some addition here, but it's growing faster on the plus end. Okay, and so the relative rate of growth on the plus end is faster than the rate of growth on the minus end, and you're actually losing some subunits there. So the overall length of this microtubule doesn't really change over time, but you can see that it's sort of moving along, and the rhodamine is sort of dye, as indicated by this sort of red triangle, is sort of moving towards the end, right? Because you're basically getting plus end growth, minus end shrinking. It's as simple as that. Another thing that microtubules undergo is this thing called dynamic instability. And I really want you guys to understand dynamic instability because it becomes really important in the cell cycle. Okay, and so what you have with dynamic instability, so oftentimes you'll have a nucleating factor down here on the minus end, so the minus end doesn't really change, but what you'll end up having is rapid growth with the GTP cap. Okay, and eventually the concentration of the free subunits in the cytosol with the GTP bound is actually going to sort of decrease to the point where hydrolysis is basically occurring faster than the addition. Okay, and when that happens, the hydrolysis of the GTP cap, you're going to have a GDP bound form at the cap, and that sort of favors disassembly. And so what you end up having is what's called a catastrophe event. okay, and the microtubule rapidly shrinks, okay, because it all has GDP-bound form in it. Okay, and then eventually the free subunits in the cytosol will get the GDP exchanged for GTP, and you'll start to regain that cap, and then the microtubule can grow again. Okay, so this is what's called dynamic instability, where the microtubule grows, and then the rate of hydrolysis is faster than the rate of addition, and then you lose the GTP cap, and a catastrophe event happens, and the microtubule starts to rapidly shrink, and then all of a sudden the free subunits in the cytosol will have the GTP bound form, and the cap gets reassembled, and the microtubule can grow again. Okay, and so catastrophe occurs basically when the rate of nucleotide hydrolysis exceeds the rate of addition. Okay, and the reason it becomes unstable is it sort of undergoes a small little conformational change. Okay, so here is your GTP-bound tubulin dimer, and it has the beta subunit has GTP, and then eventually that's going to hydrolyze the GTP to GDP, and when it does that, it creates sort of like a kink, and you get this sort of curved protofilament, and that leads to depolymerization, and eventually when it gets back into the cytosol, you'll get the GDP-GTP exchange, and then the subunit can be added back on. Okay, and here's sort of what it looks like. You have your sort of growing microtubule, and it has the GTP cap, and then all of a sudden when hydrolysis outpaces the addition, the tubulin in the GDP-bound form likes to sort of bend apart, and it starts to peel off, and you get the microtubule shrinking. Okay, so here's a microtubule with a GTP cap, and it's nice and stable, and then all of a sudden when it starts to undergo dynamic instability and shrink, you can see it sort of curls back, and this is just such a beautiful EM here. You can see it here as well, especially these are just microtubules numbered 1, 2, 3, and 4, and you guys can sort of look at the slides and follow them yourself. If you just follow number 1 here, it starts off at time 0, and then it initially starts to shrink, and then it can grow again, and then it shrinks again. So the microtubules are constantly undergoing this dynamic instability in the cell where they're growing and shrinking. And this all happens on the plus end because the minus end is typically bound by a nucleating protein called gamma tubulin, which we'll talk about in the second part of today's lecture. As I mentioned, there's a ton of proteins that interact with both the cytoskeletal proteins, and specifically actin and the microtubules, as well as the intermediate filaments. And here's just a little spider diagram of all of the different interacting factors that associate with actin. Okay, you have your motor proteins, the myosin, then there's a whole bunch that are involved in cell division, budding and polarity, secretion and endocytosis, lipid synthesis, and filament dynamics. Okay, and then there's just a whole bunch of other proteins too with sort of unknown functions. The intermediate filaments, once again, these are sort of create these sort of rope-like properties, and here you can see a nice EM of some intermediate filaments, and typically they form a coiled coil, and then those coiled coils get staggered to form tetramers, and then they get packed together, and eventually they form sort of this rope-like filament that's here. Here's just a little table of all of the different intermediate filaments. You have your nuclear lamins, and then you have some in the cytosol like desmin and vimentin, and then some ones you really should probably know are the keratins. Okay, and these happen in epithelial cells, and these are actually one of the most abundant proteins in our skin cells, believe it or not. And actually, here's a nice fluorescence image of keratin in epithelial skin cells. You can see it's sort of stained here in green, and it's found in skin and hair and nails, and it's the major protein in epithelial cells, and it provides a certain amount of strength. And if you go back to the figure I showed with the endothelial cells, it's often anchored to the desmosome, and we'll cover that more when we get to the extracellular matrix and how cells are attached to one another. But there's actually diseases if you have mutation in keratin, and this is epidermolysis bullosa simplex, and it's a mutation in keratin, and it basically causes blistering on the skin. And the point is that you have a defective keratin network, and so it's easy to sort of peel off the skin cells, and you blister quite a bit. And then there's also intermediate filaments in the nervous system, and here's just some nice scanning EMs of some of the intermediate filaments in nerve cells, and you can see here how they compare to the microtubules, and these oftentimes will help to give cells basically their structure and whatnot. So that's all I have for part A. In part B, we'll talk more about some specifics and how these filaments are nucleated and things like that. Okay, I will see you guys in a bit.